Okay, well, welcome. This is Notes from the Lab, an ongoing series of conversations on the past, present, and future of type 1 diabetes. My name is Denise Faustman, and I'm head of the immunobiology labs here at Mass General Hospital and Harvard Med School. And today we have a very special guest on this podcast, and I'd like all to be introduced to Dr. Åke Lernmark, who is a professor at Lund University and really one of the world's experts at type 1 diabetes and has seen over the years so many trends or the lack of trends in this disease. So he recently wrote an editorial on type 1 diabetes, and that really got us thinking that he needed to be a guest, a special guest on this series. So maybe, Dr. Lernmark, you could start this conversation by telling us, and I don't know the answer here, how you got involved in type 1 diabetes work. How did you select that field?
Okay, well, thank you, Denise, for having me. And I should also add that since you brought up the letter or editorial that we wrote for Diabetologia, that I should also give the best regards from Jörn Nerup, my co-author, and he
Yes.
says specifically say hello to you.
Okay, great. Well, and if
He was unable to participate.
okay, well, we'll think of him. Make
Yeah,
sure you share his opinions too.
so yes, we've been working on this since last summer and have met and have had some very interesting discussions, as you can imagine. But anyway, to answer your question, my interest in diabetes dates back to medical school at the University of Umeå more than 50 years ago. And being a summer student up in Umeå, a visiting science from the UK taught me the radioimmunoassay for insulin, and I also learned how to microdissect pancreatic islets
Mhm.
and from the mouse pancreas. And I thought the islets were complicated, and I wanted to make single cell suspensions to isolate beta cells to measure insulin secretion. And that was part of my doctorate, which is usually done when you have obtained your MD to practice. But I skipped that part and went to the University of Chicago instead to work with Donald Steiner, the guy who discovered proinsulin, and worked in his lab for more than 3 years.
Mhm.
And during the years in Chicago, where Don Steiner let me do pretty much what I wanted, we found out that children with newly diagnosed diabetes have antibodies to the membranes that I was isolating to isolate the glucose transporter in the beta cell. And we also found that these antibodies that the children have inhibited glucose stimulated insulin release from beta cell suspensions. So, since I worked in a biochemistry department, we wanted to know what the antigens were. And that work was initiated at the Hagedorn Research Laboratory, where I moved to become the director of research. So we found the first autoantigen in the children's sera, a 64K protein, but it would take 10 years before our lab at the University of Washington in Seattle, where I moved, uh, was a novel isoform of glutamic acid decarboxylase. So that was pretty exciting. And we were also then able to develop a radio binding assay for autoantibodies by in vitro transcription translation, a technique that I learned in Chicago. So this RBA, this radio binding assay, has now been the gold standard for 30 years,
That's pretty good.
which makes you nervous. You know,
Yeah.
it's only the last few years that the non-radioactivity assays have been developed. So, the early antibody methods made it possible, and also the HLA typing that we have worked on that in Copenhagen it made it possible to convince NIH that screening newborns for HLA risk and then follow high-risk children from birth until autoantibodies develop for 15 years. That was possible. So that TEDdy study took me back to Sweden, to Lund University Clinical Research Center in Malmö. And this year we finished the sampling phase. And as you can imagine, there is plenty of data to analyze to find out, what triggers the first autoantibodies. And that part is, going to involve children who have the autoimmune pathogenesis, but not necessarily the etiology. So that was a long background.
Yeah, but when I started the field, I think I remember you and one other group talking about the GAD assay and which subprotein it was and what subunit it was in many sessions. So you kind of brought back a memory for me. Of course, I just wanted to know the right answer, but you and other colleagues were really into the details of which, wasn't it GAD 64 versus 65 was the debate?
Yeah, we started with the 64 protein, and out of that, we cloned human islet GAD, and it turned out to be a novel isoform and that the people working on GAD in the brain has suspected, but we were actually the first to sequence the human GAD65 isoform, to the dismay of the brain people. They thought they should have done it.
Well, you never know in science where the little.
Oh, no, that's
you
right.
Don't know. You cannot predict. You have
to be open-minded.
Yes. So who's going to have a different answer? So maybe if we could share with the audience, over the years, there's been many theories on how and why you get type 1 diabetes. Do you want to just outline a little bit of, with your career, how people thought it occurred first and then second, third, and fourth? And I know you're going to question what we think today is the etiology. So, but maybe it'd be helpful just to go through the history of what people thought was causing this disease.
So the development of the paradigm that type 1 diabetes or autoimmune diabetes was really due to the immune system attacking the beta cells of the patient itself. It began 60 years ago. And in the 1960s, it was shown that thyroiditis was most likely, it was deemed to be an autoimmune disease. And if you injected thyroid extracts in rabbits or rats, you could mimic the disease. So it was felt that the thyroid autoimmunity had fulfilled the Cox postulates that you could induce the disease in an experimental animal. And that was attempted first and 60 years ago when Jörn Erup, he was a young medical student or medical doctor who looked at all the Addison's patients in Denmark, and those who needed insulin was recorded to have, they had Addison and they had type 1 or autoimmune diabetes. And then a colleague of Jörn found the same thing in thyroid autoimmune disease. And the seminal work that Jörn Erup did was to show that patients with the newly diagnosed type 1 diabetes had blood cells, peripheral blood cells, proliferating against islet antigens. And then it was hypothesized that insulin-dependent diabetes, as we called it those days, was an autoimmune disease. And this hypothesis was then further supported by the demonstration first in the study, both in one study in diabetes showing that HLA-B15 was associated with insulin-dependent diabetes. And then Jörn Neropad and colleagues had a major paper in the Lancet demonstrating that B8 was also associated with type 1. And then that group also showed that DR, the later discovered HLA-D related specificities, were associated with type 1. And then that was one thing missing, and the missing link was the autoantibodies.
Yeah.
And people worked on the autoantibodies for years and couldn't find them. But Franco Bottazzo and Deborah Doniak and at the same time, a Scottish group reported in the Lancet that yes, patients with concomitant autoimmune disease, they have autoantibodies against islets in frozen sections of human pancreas.
Mm-hmm.
And the trick in all this was to find out that they found out that you could only use pancreas from blood group O donors to avoid non-specific binding.
I see, yes.
So then everybody got excited, and the editor of Diabetologia decided that we need to shape up the research on the etiology and pathogenesis of diabetes, and therefore only papers calling diabetes either type 1 or type 2 were allowed to be published in Diabetologia. So,
that I did not know about.
Oh,
that
you didn't know
I
that?
did not know about. Okay.
It was very clear, and we were pretty upset, But anyway, so that is where the research has landed today, that type 1 and type 2 is so established that it is very difficult, for example, in type 2, to acknowledge that there are different subtypes of type 2, right?
Mm-hmm.
And I think the breakthrough there was the demonstration of monogenic disease. If you have a mutation in glucokinase, you can develop diabetes, should not be treated with insulin.
Mm-hmm.
So I think that's the stepping stone for dissecting the type 2 conglomerate of different diseases.
Mm-hmm.
And then, of course, type 1 was left, and with better antibody assay, sharper genetic associations with genetic polymorphisms, and the antibody assays, also detected in adult patients masquerading as type 2, but they really had type 1 diabetes, and they were called LADA, etc.
Yeah.
So that's where we are with the major hypothesis then, that what we call today type 1 diabetes is an autoimmune disease. It could have been called autoimmune diabetes.
Well, of course. And then it was called for a while juvenile diabetes too. There was for sometimes I still see that in the literature, juvenile diabetes.
Oh, absolutely. And, when I began, it was the transition from maturity onset diabetes or juvenile onset diabetes.
Mm-hmm.
And then more and more people make the distinction between being insulin dependent and non-insulin dependent.
Yeah.
But that was then dismissed by the editor of Diabetologia, who wanted type 1 and 2, right?
Well, I think a lot of people are questioning these days the authority of journals and trying to think about publishing things faster. So we'll see how this shapes up over the next 10 years as that idea of publishing gets challenged as well. So in some of your articles, you talk about the old hypothesis, just so everybody on this podcast knows what you mean. Could you just define in your words what the old hypothesis would be?
Yeah, the old hypothesis is that all type 1 diabetes is explained by an autoimmune triggering mechanism. For example, that there is one common way by which the first autoimmunity that we can measure, namely the appearance of an autoantibody, that it has a etiology that is the same, and it has to do with the subsequent development of autoimmunity against the pancreatic beta cells. So now we had a problem with this, and we sat down and said, why is it that there are so many individuals who have the HLA genetic risk? In Sweden, about 26% of the population have the HLA risk, and some of them also develop autoantibodies, but they never develop type 1. So why is that? And then, so why do some individuals with none of the current high-risk HLA, and we know from 1986 when we did the first HLA typing in patients, all patients developing type 1 in under the age of 18. All of them were HLA type, but there were 91% who did not have the HLA risk. And now when we repeat those studies, looking at HLA in newly diagnosed children and young adults, still 11% don't have the high-risk HLA. And then, we wonder, why is it a difference between boys and girls? For example, after 18 years of age, twice as many men developed type 1 diabetes than women. And then the other thing that had been bothersome for me since 1986 is that 90% of the children who are newly diagnosed don't have a first-degree relative with type 1 diabetes. And Sweden is such a small country, so when I was working here, it was impossible to do a family study. If you have 300 million people, like in the US and Canada, you can do screening of first-degree relatives easily. But in Sweden, there were not enough families with 2 people to be studied. So we have focused on the general population, and this finding that 90% of the children don't have a first-degree relative, it doesn't go away. It's the same year after year. And then we also have, observations that there is a difference. If you have a father with diabetes, why is he?
Isn't that fascinating?
He's confirming, the double the risk compared to the mother. And then, we have only 2 major HLA haplotypes. And why do they predominate in type 1 diabetes? And of course, in the TEDdy study, we see phenotypic differences between the 2. And then the most interesting study, which we were paid for by NIH to do in Swedish 15 to 35-year-olds, 15 to 35-year-old patients recruited when they were diagnosed with diabetes, that the protective HLA haplotype are very effective to protect children, but they don't protect the 30-year-olds.
Yeah.
So HLA-DQ6, for example, everybody take for granted that if you have that, you're not going to develop diabetes. But that protection is only true up until 35 years of age. So
Mm-hmm.
there's something missing here, and we are proposing that the missing link is the beta cells in the newborn.
Mm-hmm.
It cannot be excluded that if you have an inborn error of metabolism that initiates perhaps spontaneous necrosis, that may explain a sizable proportion of children who then go on to develop type 1 diabetes. It may take 1, 10, maybe 20 years before diabetes is detected, but the etiology is that spontaneous or inborn error that you were born with.
Mm-hmm.
And then it would be very easy to understand the autoimmune arm of what we call type 1 diabetes. The autoimmune arm would also be dependent on a inborn error of necrosis. And if that happens, if you are HLA DR4, DQ8, you may develop insulin autoantibodies very early because of the HLA.
Yeah.
Or if you are DR3, DQ2, you may develop GAD autoantibodies very early because of the HLA.
Mm-hmm.
So it doesn't, we are not saying that the etiology may not, what we're saying is the etiology is something else than a trigger that always end up in an autoimmune pathway. The trigger is something else, but depending on your HLA or genetic propensity, you may end up in an autoimmune pathway or you may end up in a non-autoimmune pathway. The end result is the same. You are losing your beta cells and you are going to be insulin dependent for the rest of your life.
I mean, it's pretty interesting also the twin studies. The twin studies are interesting because if you have type 2 diabetes in your two twins, both are gonna kind of get type 2 diabetes. But in autoimmune diabetes or juvenile diabetes or type 1, if you've got a twin, the other twin doesn't necessarily have to become insulin dependent or
That's correct.
yeah, so the environment or something. And then the other paradox, which you've studied too, is if you kind of are in the latter category and you
Right,
die, you got a lot of islets in your pancreas. There's a lot of
right.
islets there. Okay.
Yep.
So, like, if you have autoimmunity, why aren't those islets bumped off? Why? Why do they persist for so long in LADA?
So if you think about an ideological trigger that reduce your beta cell, it would be understandable that you live your life with a deficiency of beta cells.
Mm-hmm. Yeah.
And if there is no autoimmunity, depending on the aging, depending on the way you have grown, etc., you may, if you are physically very active, you may live a long life with a shortage of beta cells.
Yeah, because you don't get
Despite
insulin
this,
resistance. You don't get insulin.
right?
Yeah, yeah. So if all the trials, I mean, so many of the big trials, maybe more centered in the US than in Sweden, have been using immunosuppressive drugs. How do you think that fits into the changing ideas of how diabetes might be occurring?
I think the important part there would be to really understand the triggering of the autoimmunity and when to use what in terms of immunosuppression. I think, and as you know, since the very beginning with cyclosporin, cyclophosphamide,
Yeah.
everything developed that has an immunosuppressive effect or action will
Mm-hmm.
be tested on newly diagnosed type 1 diabetes patients.
It's guaranteed. It's
A
guaranteed.
guarantee. That's one thing we know it's going to happen. And so far, I mean, there are promising treatments now,
Mhm.
but having followed the different, both open and controlled trials for so long, we're not there yet. We're asking the right question with the wrong agent, right?
Mm-hmm. Mm-hmm. I mean,
And
you're absolutely true. I remember all the cyclosporin A trials and then the kidney toxicity, and then it went to anti-CD3, and then it went to anti-thymocyte globulin, and every known immunosuppressive agent has, as you said, been tried in type 1 diabetes. So that class of drugs has not necessarily resulted in a home run that everybody hoped to see, especially
right.
with safety. Safety's huge in kids, right?
So
Safe.
if we are, able to identify the ideological mechanism, maybe there is an opening for combination trials that if you treat that primary deficiency or faulty beta cells, and then the autoimmune reaction has been initiated, I think we are in a much better position to, make combination trials that may block the progressive autoimmunity.
Mm-hmm.
It's still going to be a major effort, but so far, and I think both you and I agree that we need to do something else. We need to understand the timing of the different steps. And, as in many diseases, when you come in too late, you can try most everything, it's not going to help.
Yeah. We've been overlapping a little with the Alzheimer's community, and it's kind of, they're almost in the same place in many ways that they tried every type of in end stage Alzheimer's, and none of
In
those trials,
NSAID.
billions and billions of dollars, have panned out. And in fact, the drugs themselves have safety issues. So I think that field, as well as diabetes, and Alzheimer's is obviously a complication of diabetes as well. That field has shown maybe we're going way, way too late. So it's an interesting
Yeah.
analogy.
And at the same time, I had the opportunity to know people when I moved to Seattle at the Fred Hutchinson Cancer Research
Hmm.
Center, and later they started their own research laboratory because the cancer people didn't like the idea that T cells had anything to do with cancer.
Well,
So,
yeah, exactly. Yeah,
and you,
there's a lot of history of blips along the way, right? Of
yes.
the wrong theory for a long time. So if today somebody gave you $1 billion, let's say Bill Gates comes to Sweden and he goes, Aki, I really like you. You get to run the trial. I'm going to give you the money. It just has to be in type 1 diabetes. What trial would you, if you didn't have peer review and people telling you you were wrong, what trial would you run with that money?
So I would run a trial where we would identify uh, children, not the way we did in TEDdy,
Mm-hmm.
but we would identify children in at birth and during the first years of maybe year 1 and 2,
Mm-hmm.
with the indication of a beta cell mass that may not be sufficient.
Like less during growth, like using
Yeah, and
ultrasound or MRI. and seeing
right.
them.
And that would be possible to identify that if you think about the normal distribution of the beta cell mass, that we would focus on those children who are below the 95th percentile. And the difficult part is, of course, what are we going to use? And I can think about, we know a little bit about growth factors, early growth factors for beta cells, and the many years that have been in research very successful to look at the factors which are important to the development of beta cells. I think those are the growth factors that I think would be, tested and ask the question if that is something that will alter the way the beta cell distribution looks like in these children.
I mean, it'd be interesting, right? Because most people worldwide have not focused on the pancreas; they've focused on the T cell,
Right.
the T cell,
and then, of course, as a second arm, I'm still fascinated about the possibility because I did experiments when I was still working in the lab to make newborn rabbits and newborn rats tolerant to different autoantigens. And this is work that goes back to the 1950s in Australia, where it was shown that if you injected the newborn with an autoantigen, you could make them tolerant for life. And that's, a technique that we actually use to make newborn rabbits tolerant to the glucagon receptor. And then we can make, antibodies without the rabbit reacting to
the antibodies.
To the and so I if that kind of work will eventually, and we do all kind of things with newborn babies, to prevent them from developing disease later in life, not at least blood transfusions, etc.
Yeah.
So why not induce immunological tolerance very early in the newborn?
I think Russell's going to say we need shorter time here, but remember allergies. There's for the people in the allergy field, in the U.S. for years, for 20 years, if you had a baby, you were told, oh, feed them one group at a time for 3 months, and then feed them another food group. And then all these kids ended up with allergies. And everybody goes like, why do all these kids have allergies? And that, has dramatically changed over here. And now it's like you have a newborn, feed them anything they'll eat. Like feed them every food group, feed them peanuts, get them tolerized at a really early stage to a lot of
give them peanut powder.
Yeah, exactly. Gluten, everything. And so that's really similar to what you're saying about autoimmunity. You need those exposures.
We need to work that out.
Yeah. Well, it's so nice talking to you today. It's really a pleasure to get caught up. And once in a while we overlap at meetings, but it's always so fast and so brief. And this podcast, I'm sure, will be watched by many people. So we'd like to thank today Åke Lernmark for joining us, and certainly all your contributions to diabetes research over the years. It's really quite a journey. It's a journey.
Yeah, it's a journey.
It's not for the mild of heart. And it's wonderful that Dr. Lernmark wrote this editorial, just bringing up the idea that we may not know everything about type 1 diabetes. And maybe over the last 20, 30 years have been focused too narrowly on just one or two hypotheses. So I'm glad for your bravery. I hope there's not too many editorials to the journal editors. And thank you for your contributions to diabetes work. It's sincerely appreciated.
Thank you for having me.
Yeah, no problem.